My name is Katrin Lecture. I'm working at the British Antarctic Survey as a senior biodiversity scientist, and today I'm giving you a lecture about gastropods from the Southern Ocean hydrothermal vent fields. So on the map here you see the Southern Ocean and you see the Scotia Sea, and on the right hand side we have the Scotia Arc with the South Sandwich Islands and this East Scotia Ridge Arc. So in 2009 we found that there were evidences of two hydrothermal vent systems, the black smokers with red dots at the segment E2 and E9 and the Camp Caldera in the yellow dot. So from there, when we went on an expedition with an RV, we were able to find the hydrothermal vents. But when we saw them, we realized we had noticed those images before, because in February 1966, the American research vessel El Tanin was out on a geological cruise with a drop-down camera, taking photos whenever the camera's logger touched the sea floor, and we had an exposure. And this black and white image shows you a site in 2,377 meters near the South Sandwich Islands. In January 2010, more than 50 years later, the RSS James Cook came to the same site. And what did we discover? We discovered a hydrothermal vents area. So the next vents, this is the off diffuse flow sites and looking similar in the same animals as in 1966. If this would have been published, the first vents on the world would have been discovered in the Southern Ocean, but nobody knew what these animals were and how significant their find was. So our East Scotia Arc Ridge vent sites were significant. They were magnificent. Here you see our site black and white. So on the left-hand side, we see a photo mosaic of black and white. And black and white is because on the left-hand side, we have a darker area. We had a black smoker. On the right side, we have an area with long-stalked barnacles. And from this photo mosaic, we looked into the assemblages and saw which animals were most important. We had the white kiva crabs, yeti crabs standing out. We had dark brown areas with a gastropod. We had slightly orangey yellow ones where we had the long stalked barnacles. And then we had the big anemones outside in the helofauna. And so you can see in the colored images how defined the vent sites are by the assemblages. And you can see gastropods were important. And this is where I came in, because gastropods are one of my expertise. So we went in, and from the site, we discovered 5 different species. All of the species were new to science and were described between 2015 and 2020. So you see, it takes time to describe a species. And in the next slides, I will give you a bit more information of each species. But before we analyze the food web, how do we know that they were depending on the hydrothermal vents? This was done by analyzing the tissues for stable isotopes, for carbon, nitrogen, and sulfur, and then we looked at the ratios. And here you can see our 3 sites we analyzed. At the top, E2, E9, and then the Camp Caldera. And on the right, you see the 3 different species we analyzed, and all clearly showed in the food web that they are depending on the chemosynthetic ecosystem and the bacteria and the diffuse flow. Coming to our first species that I want to come into a detail, it's the little limpet Lepidodrillus concentricus. On the left, you see where it lives. Either it sits on the kiva crabs, on the basaltic rocks. At the bottom, we have the more unusual sites, because bottom left, this is a pure sulfur chimney. And on the right-hand side of that panel, you have basalt in a diffuse flow. And on that, they also sit on little sea spiders. When we look at the middle panel, the black one, where we have 4 sets of shell pictures, we thought initially we had 3 species because their morphology was so different, especially the ones that are higher than wide. And then we realized they were higher than wide in the Camp Caldera because they started off living on the legs of sea spiders and they were size restricted there. So on the bottom you see the kiva crab where the limpets live on and graze on the bacterial mats. On the right hand side you see scanning electron microscope photographs of the shells. The juvenile shell that tells us that this animal comes from a planktotrophic species, a species that releases larvae in the water column. Then the soft parts and the radula that give us information if a species is different, because each species often has a very distinct protoconch, soft parts and radula teeth characteristics. We also looked at their genetics, and one is to looking at the phylogenetics to see how your new species compares to the familiar ones, the same species in the same genus, and our one clearly was different to all known Lepidodrillus species, with the closest relationship to the one from the Atlantic, the Lepidodrillus atlanticus. And when we looked at the population genetics, comparing many specimens from our 3 sites, we realized they all clustered together, but there was a little bit of a difference between the Kemp caldera and the other vent sites, but the currents did their job and connected them. Provanna cookie is a small, about 1 centimeter tall gastropod that we found on the basalt and near the bacterial mats. We often found it in diffuse flow areas, not really with the high water temperatures. And you can see in the black plate where we have the different animals, we have various different growth types. We have a large ones with radial spires, we have quite flat and round ones, we have little black ones. And it was again the genetics that told us that all these growth forms and different looking animals were the same species. But what they have is actually, they have precipitate around the shell. The normal one, they have a very thick periosteum, the organic layer that's above the carbonate layer, and then the other ones have either orange or dark black precipitate. In the Brucella indurata, which is a tiny skinny-eat gastropod that is only 4 or 5 millimeters in size, we had various specimens, but we only had one that could we really identify as a trochiform animal, which is the one on the top with the left, the A specimen. While when you look into the bottom right, that almost doesn't look like a snail. It almost looks like a cecum, just a worm-like feature. And what happens here is that they live in the acidic water of the diffuse flow, and all their shell is corroded away. But the little snail still manages to keep a shell to be able to survive. Coculina enigmadonta was found in the white smoking area of the Kemp caldera. We found it on basaltic rock. We found it in the areas of the white chimneys. We found it in an area we call Toxic Castle, because almost no life apart from these snails and sea spiders were alive. And we also had a whale fall where they lived on. And so far Coculinids had only been found on woodfalls, but not on hydrothermal vents. So this was really exciting for us. And when we looked at the shells, and you see that in the black plate, only the bottom left one has a little spire sticking up, like a typical limpet shape. The others are quite flat. And again, what happened here is that the periosteum got disturbed, and the acidic water was able to come in and dissolve the shell. And the animal is since then trying to rebuild shell material so that they don't have a gaping hole in the middle of them. So with the Coculina enigmadonta, over the years we also developed our skills, because now we didn't only took the scanning electron microscope photographs and the photographs of the shells and the genetics, we also then looked at soft parts and cut them with histology in tiny hundreds of thousands of slices. They were photographed and then put together as a 3D image so that we can actually see inside the animals and see how the different organs are looking. And this is where we come to the star of the show, the Gigantopelta chessoia. Yes, it's a giant for its family. It's about 4.5, 5 centimeters in the largest, but all its relatives are only about up to 5 millimeters. That's why they are large. And we have a little video that plays, and you see here a huge community assemblage of the Chessoia, Gigantopelta chessoia. They are living in diffuse flow waters, and they are sitting there on top of each other. And we had up to 20 centimeters just filled in with these snails, one on top of the other. You see some limpets in the area, you see a little sea spider, but you also see juveniles. And when we collected them, we often had little juveniles sitting very close to an adult or in the opening of an adult snail. So this was great. We looked at them. We found at the same time when there was an expedition to the Southwest Indian Ridge, and they found a very similar looking snail and one that had scaly foots. The famous scaly foot gastropod, also a peltospirate, also large, but the two are actually not very related. Our Gigantopelta was a new genus with two species, one in the Southern Ocean, one in the Southwest Indian Ridge. And so we had a closer look into it, and there it is, the 3D anatomy of the animal. We have the different nerval system, we have the esophagus gland shown in tiny brown here, which is important when you digest your food. And we had so many animals, from tiny, tiny ones of 2 millimeters to the 4.5 centimeter ones. So we again did the histology, cutting slide by slide and reassembling them. And what figured out is that the small specimens, the esophagus gland is really small, but when they grow, especially when they are over about a centimeter, it enlarges significantly. It enlarges so large that it's up to 1,000 times larger than in other gastropods and other animals. So this is why it was called now a trophosome. And this trophosome is filled with bacteria. So this large animal doesn't need to graze anymore. It just needs to be in the diffuse flow water so that the diffuse flow water reaches the bacteria in that gland, and they have their own factory for the food in themselves. So this was a big find. And then we went back to the site, which we first saw in 2010 and then looked again in 2013 and in 2019. And what did we find in 2019? Nothing. Everything, the entire site at Cindy's Castle had disappeared. The diffuse flow had disappeared. So the hydrothermal activity had shifted slightly. There was no heat coming through anymore, and the whole community disappeared because they didn't have the water that would feed the bacteria in the gastropod, so it died off in this site.